Hey y'all, Professor Hurley here with another episode of Purple Bear Biology. On this channel we discuss anatomy and phys, microbiology, and some general principles of biology. If you haven't seen my other videos, be sure to subscribe and explore the cool world of these topics. In this video, we're going to focus on one topic, the neuromuscular junction. I know what you're thinking. The neuromuscular what now? What we are really talking about is how our nervous system stimulates muscle contraction. This interaction occurs at an articulation site between motor neurons and the plasma membrane of muscle cells. The easiest way I've found to discuss this topic is to draw it out step by step. So that's what we're going to do. This video is going to take you step by step through the process of a neuron stimulating sarcomere shortening. The video picks up where our last conversation about muscle cellular structure left off. What? You haven't seen that video yet? Oh man, it is super cool. You should watch it first. I'll put a link in the description section below. So let's go ahead and get started with this material.
So let's get started. First, I'm going to draw the sarcolemma of a muscle cell. Here is the extracellular fluid, and here's the intracellular fluid. Embedded in this membrane are two protein types, ligand-gated sodium protein channels, and these are referred to as voltage-gated sodium protein channels. Ligand-gated channels open when a chemical binds to their active site, and voltage-gated channels open when there's an electrical charge change within the cell. This elongated channel is referred to as the T-tubule, and then this box at the end is the sarcoplasmic reticulum that we're going to abbreviate as SR. Inside of the sarcoplasmic reticulum, calcium is stored. Connected to the plasma membrane of this muscle cell, we have a neuron. This is referred to as the synaptic end. Inside of the synaptic end, we can find vesicles that house chemicals called neurotransmitters. Now, there are a lot of different types of neurotransmitters, but the one that gets released at skeletal muscle cells is called acetylcholine. We abbreviate this as ACH for short. This entire structure here is referred to as the neuromuscular junction, or NMJ. Now that we have all of our structures organized, let's talk about how we get contraction. First, a positive electrical signal is sent down the neuron to the neuromuscular junction. When this message reaches the synapse, it results in ACH being released into a small space called the synaptic cleft. ACH then binds to these ligand-gated sodium protein channels. Because this channel is receiving a signal from a neurotransmitter, we refer to it as a receptor. This specific receptor that receives ACH is called a nicotinic receptor. Receptors and neurotransmitters are specific to one another, kind of like keys to locks. Once the protein channel is open, sodium, which is actually in higher concentrations outside of the cell than inside of the cell, floods into the cytoplasm. The accumulation of sodium in the intracellular fluid leads to an electrical charge change from a negative charge to a positive charge. These voltage-gated sodium channels recognize this charge and open to allow additional sodium to flow in. As sodium begins to accumulate on this side of the membrane, the charge is passed down, causing additional channels to open and allowing more sodium to flow in. Effectively, this has a cascade effect where the positive charge gets passed along the membrane and down the T-tubule. When the positive charge reaches the sarcoplasmic reticulum, sodium flows in just like it did along the rest of the membrane. Attached to the sarcoplasmic reticulum are voltage-gated calcium channels. As the charge changes, these voltage-gated calcium channels open and allow calcium to flow into the muscle cell. The calcium then travels to the sarcomere, where we have thick filaments called myosin with heads, and thin filaments called actin with active sites. Recall that actin has regulatory proteins wrapped around them. These two regulatory proteins are tropomyosin and troponin. When calcium binds to troponin, it pulls tropomyosin away from the active site of the actin filament. The heads of the myosin filament can then attach to the active site and form a cross bridge. Myosin heads with energy harvested from ATP can then pull the filaments closer together, shortening the muscle and causing contraction. As long as our nervous system is stimulating the muscle fiber and ATP is available, contraction will persist. To cease contraction, the neuron has to first stop sending an electrical signal down for the releasal of ACH. Some of the ACH gets reabsorbed back into the neuron. The remaining ACH is broken down by an enzyme called acetylcholinesterase, abbreviated as ACHE for short. ACHE functions by breaking down acetylcholine. Once ACHE breaks down all of the acetylcholine that is left over inside of the synaptic space, these ligand-gated sodium channels will shut off. Then a sodium-potassium pump is used to pump the sodium back out of the cell. As the sodium leaves, the positive charge is reduced and the negative charge is restored inside of the muscle cell. This shuts off these voltage-gated sodium channels and has a cascade effect, creating a negative charge throughout the plasma membrane of the muscle cell. This negative charge gets passed down to the sarcoplasmic reticulum and results in the calcium channels being shut off. This means that calcium is no longer present near the sarcomere, and so troponin moves tropomyosin back over the active sites, and relaxation takes place, and the sarcomere elongates.
Now that's some crazy cool stuff, but how do we make sure we understand all of this? Well, let's apply it to a couple scenarios. Let's start with something called Botox. If you've ever heard of Botox, it was probably in reference to trying to smooth out wrinkles on the face.
This chemical used is actually a toxin produced by Clostridium bacterium. Outside of Botox, this bacteria can be found in improperly canned home goods. If consumed in large quantities, the results can be fatal. But why? What effect does it have? Let's jump back in to the neuromuscular junction to see its effect. Botox works by inhibiting ACH from being released into the synaptic cleft. So how do you think this affects contraction? Well, if no ACH can get released, then we cannot get sodium to pour into our muscle cell, which means that the voltage-gated sodium channels do not get activated, which means that calcium cannot be released out of the sarcoplasmic reticulum, which means that the myosin heads cannot attach to the actin filaments, so we don't get contraction. That means no electrical stimulation results in contraction, and we get the inability to contract. This is referred to as flaccid paralysis. Let's consider another example, nerve gas. Nerve gas inhibits ACHE, so what do you think would be the effect that it would have on muscle contraction? Recall that ACHE breaks down acetylcholine, so if it's not working correctly, then we get a buildup of ACH inside of the synaptic cleft. This results in massive amounts of sodium pouring into the cell, which gives us a large electrical charge, which stimulates these voltage-gated sodium channels to allow sodium in, which passes that electrical charge along the membrane down the T-tubule, resulting in sodium flowing into the sarcoplasmic reticulum. This causes calcium to flow out near the sarcomeres, allowing the regulatory proteins to move out of the way and giving us sarcomere shortening so that we get contraction. But the problem with nerve gas is even if the neural stimulation for contraction stops, there's an excess of acetylcholine that cannot be broken down inside of the synaptic cleft. This means that contraction cannot halt and that calcium will continuously be released. Interestingly, this produces a form of paralysis as well, except this is called spastic paralysis, where your muscles cannot relax. Let's use one more example, rigor mortis. Rigor mortis is something that happens to our bodies shortly after we pass away. The body becomes stiff and rigid. So what happens exactly? Well, what happens is that the sarcoplasmic reticulum when we die no longer holds on to calcium, and it can free flow into the muscle cell. An excess buildup of calcium inside of our muscle cell next to our myofilaments results in cross-bridge formation. Now, when we first pass away, there's some ATP present inside of our muscles, but we don't have any more to make after we have died. This ATP can be used to form cross-bridges between the active sites and the myosin heads. However, once all the ATP is used up, no more active cross-bridges can be made, and the cross-bridges cannot be broken. So now that the muscles have contracted due to an excessive amount of calcium, but have now run out of ATP, they cannot relax. So this gives us the rigid nature of rigor mortis. Now eventually, after being dead for a period of time, these fibers will actually start to break down, and so relaxation occurs again. This is one of the cool ways that we can determine the approximate time of death up to 36 hours. If the individual has died less than 3 hours, rigor mortis has not had a time to set in, and the muscles will not be stiff. However, from the period of 3 hours to 36 hours, the body then becomes stiff and rigid after cross bridges have been formed and all of the ATP has been consumed. After the 36-hour mark, the muscle fibers begin to break down and the body becomes flaccid again. Kind of neat, right?
Isn't the neuromuscular junction so cool? Who knew that sodium, calcium, and neurotransmitters could have such a profound effect on our ability to function properly? If you found this to be as cool as I did, click the like button and subscribe so we can continue to explore these awesome concepts. That's it for this episode of Purple Bear Biology. See y'all next time, and thanks for stopping by.